Bismillah ar-Rahman Rahim. This is Biology 9700 we are discussing, and we're going to be discussing the paper 2s now. And this is a November 18 paper double 2. Of course, there are different variants 2.3 and 2.4, but usually they're very similar. So every year I'm going to take one paper and discuss it. And inshallah, hope to finish them in a week or 10 days so that you can have access to these papers and you can revise your AS level biology. So, this is paper 2 which is going to be discussed now in this video. We start the first question. It says figure 1.1a is a photomicrograph of healthy lung tissue. So this is the healthy one, and this is the one which is suffering from COPD. So we've got A and we've got B. So COPD is the B one and the healthy one is A. Now when you read a question, you need to be careful and looking at it very carefully. So 1.1b is a photomicrograph of lung tissue from a person with emphysema, a chronic obstructive pulmonary disease. The image, so this is, sorry, this is A is the healthy one and then B is the one. So this is the B one and this is the A one. So B is the photomicrograph of a person's COPD emphysema, and the images are both at a magnification of x40. So they've given you the magnification of it, and now they want you to understand and see if you can answer the next question. The first question was name the structure labeled H in figure 1.1a. Now, as you can see in 1.1a, H is what? It's amongst the alveoli. It's something which is amongst the alveoli, and so it has to be nothing else but the bronchiole. Now, you're not sure which bronchiole it is. Is it the terminal or the respiratory bronchiole? I'll just revise this in a minute. And then it says calculate the actual diameter of xy. So here is the question, is the calculate the actual diameter of xy. So then you needed to measure that and then calculate the actual diameter of that. Now, how will you do that? Now, this is always a problem with you all. I don't know where we go wrong as teachers. I blame myself. Is that you've never understood this magnification. Some of you just refuse to understand it and sit down and make an effort to understand it. So if you look at the formula, it is the actual is equal to image over magnification. Now the image is, as you can see in it, is 1.2 centimeter, which I have given you. Now 1.2 centimeter is equal to 12 millimeter, and that's equal to 12,000 micrometers, because if you look at the answer was expected in micrometers. And then you divide it by the magnification. The magnification given in the question was x40. So the magnification 40, you divide it and that's 300 micrometer. That's the actual size. So the actual size is 300 micrometer. Why have we to give it? Because this unit was given to us in the question that we had to give the answer in micrometers, not in nanometers. If it was asked in nanometers, then we would have said, okay, 12,000. So three for the micro and then 3 for the nanometer. But no, it wasn't asked. Nanometer was not asked. The answer was asked in micrometer. So please understand this at this point and just pause and do the working and explain it to yourself. The formula actual is equal to, we have to find the actual. It says calculate the actual diameter. So actual is equal to image over magnification. I is for image and M is for magnification. The magnification was given in the question and the image was, which was of course the drawing, which is 1.2 centimeter. The C part of the question says gas exchange in the lung occurs in the alveoli. So this is the information they've given it to you. Describe the process of gas exchange in healthy lungs and suggest how this compares with gas exchange in the diseased lung shown in figure 1.1b. So please read the question twice, underline the keywords, and then think, look at the number of marks which we have to reply and how many points you have. So if you have a 4-mark question, well, then you should have at least 6 points, because maybe one of the points you haven't worded very correctly and you don't want to lose marks on that. So you write more. There's no negative marking in these questions. So you write more. So even if 2 are irrelevant, I said irrelevant, then of course you're not going to be, no marks are going to be deducted. But if you've given me the 4 points out of the 5 which are given in the mark scheme, now I will give you your 4 out of 4. So please be careful in answering these questions, understand the question, and then start writing your answer. And please think in points. So 1 point, 2 point, even if you don't put these dots, but think in points. Okay, now I've got to answer, I've got to get 4 marks. So it must be 4 very, very important points. And the 4 points should not be just repeated in another wording. Some of you write the same thing, but you just word it differently and you think it's a separate point. Well, that is not the case. It's not a separate point. It's the same thing, but you're just saying it in other words. Now, as we read on this question, woolly foxglove, digitalis lanata, shown in figure 2.1a, and common oleander, Nerium oleander, shown in figure 2.1b, are plants grown for the attractive flowers that they produce. Both plants are poisonous as their leaves produce toxic organic compounds known as cardiac glycosides. Cardiac glycosides have a powerful effect on the cardiac muscle. Read the question carefully, underline the keywords, and as we proceed, N. oleander is able to grow in very dry conditions. The leaves have adaptations to reduce water loss by transpiration. State the term used to describe such a plant as N. oleander that has adaptations to allow it to grow in conditions where water is in short supply. So the answer to that is xerophyte, and it said the reason why you're not going to get a mark. If you wrote cactus, cactus is a plant. Now it didn't say name a plant. That's not even in your syllabus. It says state the term. So when it says state the term, so then you have to be careful that what are you going to write? You're going to say xerophyte or xerophytic plants. Then it says if it's a small insects that feed on plant fluids using piercing and sucking mouth parts, and when if it feed on the sap present in the vascular tissue of leaves and stems, a sugary liquid, sucrose, amino acid, sugary liquid called honeydew is passed out of the gut. The honeydew can be analyzed to find out what is present in the sap. Now state the name of the vascular tissue from which the aphids feed. We all know it is the phloem, because if you look at the stem, the phloem is on the outside. So they suck it, and that is why I always say the phloem is on the outside, so facilitated the aphids to suck up the fluid, which is of course got sucrose in it. So it's very sugary, it's very sweet. Sucrose is the sugar that we have at home and which we put in our tea or coffee. So this is the clue that it had. It had to be phloem. It couldn't be xylem. Xylem would have water and ions, and that would be wrong. So this is what we had to have. Then it says, now as we read the further question, an investigation found that aphids feeding on D. lanata produced honeydew containing cardiac glycosides. Suggest why cardiac glycosides were present in the sap from the vascular tissue. Now, an investigation they found that aphids feeding on D. lanata produced honeydew containing the cardiac glycosides. Now, this question was given to you. This was given in the earlier part of the question that these are present in the leaves. So suggest why cardiac glycosides are present in the sap from the vascular tissue. Now the reason is that in the phloem, it's always translocation. So you have to sort of connect the phloem part with this. So translocation is what? Source to sink. And source, you give a little more about the source. Source is the place where it is synthesized, and sink is where it is required or where it is sourced. So basically the flow in the phloem is from source to sink. So we all know that, and you gave that information. I always say is, exam skill is a very important thing, that you just look at it and they're asking you what the question is, and you give a little bit more information about that part of the syllabus which they're asking you. And then we could, of course, have said as a defense mechanism against sap feeders. You see, if these insects die, then of course they won't take more of the sap, and this will save the plant because the phloem contains all the nutrients which is going to transport to the roots, because the roots can't photosynthesize. So the roots are the sink and the leaves are the source. So in a way, it would be a defense mechanism. That, of course, I would not expect many of you to come up with that. I don't think I could have thought of that in the time of the exam as well. So these are the information that you needed to give is why is it present in the sap? And then, of course, they're talking about the phloem because we just talked about the phloem here in this part of the question, and this is part one and this is part two. So remember to be answering the question as you see the flow of the question, and that is a very important skill that you need to understand. Look at this question: is cardiac glycosides have an effect on the movement of ions into and out of cardiac muscle? The outcome is an increased ability for the cells to contract. Investigation into the action of the cardiac glycoside oleandrin extracted from N. oleander have shown that it acts to prevent the correct functioning of sodium potassium ATPase, a membrane transport protein. Don't get upset by the word or the names used, but just look at what is the saying, a membrane transport protein. So those channel proteins which are found in the cell membrane. So sodium potassium ATPase has a role as an enzyme, a transport protein. ATPase is an enzyme that catalyzes the hydrolysis of ATP to ADP. So basically ATP is going to be converted to ADP and in all. So this bond is going to break between the, so we're going to have an adenosine triphosphate. 1, 2, 3. Triphosphate is being converted into only 2-phosphate. Only this is going to be left back. So ADP, so ATP to ADP, an inorganic phosphate. This PI that I have written is an inorganic phosphate. And energy released from this hydrolysis is used to transport sodium ions out. Energy. Explain what is meant by the hydrolysis of ATP using water, because here you have to add a water molecule. You see, here you have to add a water molecule because the water is going to break up into H ions and OH ions, and that is going to add to it. So using water to break bonds between the phosphate group. Now this was the 1 mark scheme point, this is the 2nd mark scheme point, and this is a 2 marks question. Then it says name the type of transport mechanism involved in the transport, and that of course is active transport. Part 3 of the question says oleander is a non-competitive reversible inhibitor of ATPase. Describe the mode of action of oleandrin and explain. So describe and explain how this will affect ion movement through sodium potassium ATPase transport proteins of the cell surface membrane of cardiac muscle cells. Now if you look at this question, which is the part 3, describe non-competitive reversible inhibitor. Now that was in the question. Now the first thing about a non-competitive inhibitor is binds to a site other than the active site. So this is the active site. It's going to bind somewhere here to a site other than the active site. And this of course changes the shape of the active site. So the shape of the active site changes. So now substrate can no longer bind to the active site. Now what do you write? You said substrate cannot bind to the enzyme. Now that's wrong. That's wrong English, because the substrate cannot bind to the active site of the enzyme. The substrate doesn't have to bind somewhere here. It has to bind to the active site in order to catalyze the reaction. So substrate, enzyme substrate complex, then there will be what? There will be no, or there will be less hydrolysis of ATP. So the ATP breakdown, this word breakdown is not allowed. So the hydrolysis of ATP breakdown is allowed. Sorry, it says in the mark scheme allowed. So hydrolysis of ATP, so less energy released. Either you said this or you said this. And then sodium is not going to be moved out. Sodium moved out and potassium will not be moved in. Of course, this was given in the question. So if there's going to be no active transport, what was going actively transported? Sodium was actively transported out and potassium was transported in. This was an active transport, and they gave that information to you in the question. Then part D of the question, it says digoxin is a cardiac glycoside. So digoxin is a cardiac glycoside extracted from the leaves of this plant, can be purified and used as a drug to treat some heart disorders. Examples of these heart disorders are atrial fibrillation. Atrial fibrillation means that you remember the heart. So it's got the right atrium and it's got the left atrium. So atrial fibrillation is when the normal rhythmic cardiac cycle is disrupted. Like for instance, the heart beats at 80 times per minute and starts to beat at, the atria start to beat at 110 times per minute. Now that would be called atrial fibrillation. And the second thing that they said was heart failure, where the cardiac muscle is contracting weakly. Weakly means not weekly or monthly, but weekly means that it is going to be working it sort of is supposed to pump out say 50 ml of blood, but it is unable to pump that out. It's only pumping out 30 ml or 40 ml. So is contracting, but it is not very strong. Weak means not very strong. Now it says describe the sequence of events, and they have underlined sequence of events occurring in the left side of the heart during one normal cardiac cycle, and include reference to blood pressure changes, and the first event in the sequence is described for you. So they help you to start off the answer to this question, and then they expect you to complete this answer. Now if you look at this question, I mean the first event in the sequence described for you, and you have to give the sequence what was occurring on the left side of the heart. Now you know the left side of the heart has the bicuspid valve and the right side has the tricuspid valve. And I usually tell you to remember it, left side bicuspid valve, LBW, leg before wicket in cricket. So the first point which was given to you is the left atrium fills with blood during relaxation of the left atrium and left ventricle. So atrioventricular valves open, atrial contraction, blood flows to the ventricles. So here, so the blood is going to flow into the ventricles, and then the bicuspid valve closes. Now, of course, this valve has to close between the atria and the ventricle, and the semilunar valve opens, which is going to then allow the blood to flow into the aorta. So the blood flows into the aorta because we're talking of the left side of the heart, and when we are talking of the left side of the heart, you know the left side, the aorta arises from the left side of the heart. And then atrium in relaxation during ventricular contraction. When the ventricle is contracting, the atria is relaxing, chilling out. And then the contraction of the atrium or the ventricles results in a pressure increase in that chamber. So we had how many marking scheme points? 1, 2, 3, 4, 5, 6. And you have to give me any 4 out of these. So any 4 and you would have got your 4 out of 4. So you've got to remember is that you could write in such a manner that you could think that you've got to get 4 marks out of this. So how are you going to get your 4 marks? And you are going to be sensible to write the correct point so that you get your 4 marks. Part 2, which is the last part of this question, this was an 18-mark question, which is a pretty tough question as far as I can think. Suggest how the health of a person with heart failure can be improved by treatment with the drug digoxin. Now everything, all the information is given in the question. What did digoxin do and everything. You can read the question again and then go back and then come back here to answer this part of the question. Now, of course, as we see how the health of a person with heart failure can be improved. Now, more powerful contraction of cardiac muscles. Now, this is 1 mark scheme point. And then blood, if there's more powerful contraction, you know the blood will be pumped at a higher pressure. So more force exerted on the blood being pumped out. So blood pumped at a higher pressure, then more force to overcome the resistance in the blood vessels. You see, when the blood is flowing in the blood vessels, offering resistance to the flow of blood. Then more blood reaches the lungs. You know, it is the pulmonary circulation and the systemic circulation. So one goes to the lungs and then going to the body. So when more blood reaches the lungs, of course, we're going to have more oxygenation. So more blood reaches the lungs to obtain oxygen. Or you could say more oxygen reaches the body tissues per unit time. So this, of course, will result in increased energy. Because you see, oxygen means more aerobic respiration, more energy released. So this is going to cause less fatigue, and then the thing and can be more mobile. The person will not feel tired and just keep on sitting all day. So increased mobility. So this was the sort of question which checks how well, number one, you've understood the question, the information given in the question. Now, how can you put it into words? So please read the question again, go through the mark schemes again, and analyze and see where you went wrong. What did you write when you didn't look at the mark scheme? You should have done this question without the mark scheme and then checked with the mark scheme. Please do not look at the mark schemes immediately, because that does not help you. That means you're just trying to just go through the mark scheme, but you haven't taken time to do the question yourself and understand the question and see how much of the answers that you wrote were correct. Come to question 3. The response of the human body to tissue damage depends on the types of tissues involved. Epithelial tissue, liver tissue, and cardiac muscle tissue each respond differently to damage. Epithelial tissue of the gas exchange system contains stem cells. Liver tissue contains cells in a non-dividing state that can enter a cell cycle when stimulated. And cardiac muscle tissue contains cells that cannot divide at all. Damage is permanent and is associated with scar tissue formation. So all this information is given to you in the question. You need to read it, you need to ponder upon it, you need to process it, and then of course you need to answer. So explain the importance of mitosis in the repair of damaged tissue. 1. Mitosis, repair of damaged tissue. Remember, I've always said to you, repair of damaged tissue, replacement of worn out cells. These are two totally different things. Repair of damaged tissue, replacement of worn out cells. I mean, your shoes need to be replaced. Your school shoes have gone to, they're torn or something, so you need to replace them. But repair of damaged tissue, you had an appendectomy, so your skin was cut. Now that, of course, is the damaged tissue which is going to repair and heal, and you're going to get a scar, maybe or maybe not. Then explain why stem cells are important in tissue repair. You can see in the A part, explain the importance of mitosis in the repair of damaged tissue. Look at the mark scheme. Produce new cells, or if you said replace damaged cells. Then mitosis results in new cells which are genetically identical. And then all the new cells can retain the function which the damaged tissue is not affected, because now that the new cells will replace it, it will retain its function which it was originally doing. Now in the B part of the question it says explain why stem cells are important in tissue repair. Now the cell cycle is continuous. It's continuously cells keep on dividing. Then this of course results in cells that can differentiate and specialize in that specific function which that tissue which has been damaged is repairing, so they can then perform that same function which they were originally doing, and then can produce tissue that can still function. So cell cycle continuous, produce cells that can differentiate or specialize, and the third point was can produce tissue that can still function. So these were the 2 marks, and there were 3 mark scheme points, and any 2 of these points would have got you your 2 out of 2. Following liver tissue damage, chemicals are produced and released into the circulation, and these chemicals are able to stimulate the liver cells to help tissue repair. Explain how this is an example of cell signaling. So if you know cell signaling, you can understand. So following liver tissue damage, chemicals really produced and released into the circulation. So it goes into the blood, so the blood is going to carry it, and these chemicals are able to stimulate the liver cells. So read the question carefully and then think of the answer. Now you see the marking scheme answers are very, very typical. Chemicals are, now this was in the question, chemicals are cell signaling molecules. You could have picked that up from the question as well. Now liver cells are the target cell, that's where they have to go to. Then the binding of chemicals to the receptors. You see the cells have receptors on their surface, and the shape of the receptor is complementary to the signaling molecule. So that's going to fit on it, just like the active site of an enzyme where the substrate fits. So liver cells are the target cells, binding of chemicals to receptors. Then specificity of receptors. You see this cell signaling molecule will only attach to the receptor on the liver cell. So that is why they are specific, because of the shape of the receptors, which is complementary to the shape of the signaling molecule. Then response is that cell enters mitosis, because we talked about that earlier on, that the liver cells help tissue repair. So tissue repair can only take place if there is going to be mitosis. So you could have picked that up from the question again. And then of course if you knew about the enzyme cascade, or if you knew about signal transduction, and if you wrote about that, you'd probably get another mark. But you had enough points. You had about 1, 2, 3, 4, 5, 6 points. You only have to give me 3 points to get the 3 out of 3. So of course this is a very large marking scheme. Some questions have a very for 3 marks it could be 6 points. Sometimes for a 3 marks question there's only 3 points, and they expect you to give those 3. Now that's a very narrow mark scheme, and those are the ones which are very difficult questions. Now we come to question 4, and this viruses share common structural features. Some viruses such as human immunodeficiency virus, HIV, also have an outer envelope as part of this structure. Outline the key structures of viruses. So number 1, they have a protein coat, and inside it either it has a DNA or it has an RNA strand. So if you said nucleic acid, you got a mark for that, but then if you specified DNA or RNA, then you got another mark. So because if somebody didn't know DNA but knew the word nucleic acid, well, I'd give them a mark for that. And then we say it is acellular. It doesn't have it's not a cell, because there is no cytoplasm. Cell is a unit of life, and cell means it has to have, number 1, a cell membrane, number 2, cytoplasm, then it's got all the different things in it, like bacteria are unicellular. But this is not, it doesn't even a cell. There's nothing in it except there is this protein coat. This outer, this is called the protein coat, and inside there's nothing. It's only a DNA or an RNA strand. Either this is DNA or it is RNA. So if it's either DNA or RNA, that's all. There's nothing in it. There's no cytoplasm in it. Please remember this. Now, as I've got to hurry up this question, calculate the percentage of pregnant women living with HIV that received HRT. So you have got a figure between 64 to 68%, and I'm sure you can figure that out in this. Then it comes to the part 2. Describe the trends shown in figure 4.1. Now, as you look at it in the trends, now when you look at the trends, generally you've got to give very general comments on it. Is there an increase? Is there a decrease? Is there an anomalous reading? And you always give me some data to support. Data means you give me figures from the graph which is given above in the question. So decrease in the total number of women living with HIV and increase in the number of women living with HIV receiving ART. So this whole thing is going to be 1 mark. Then number 2 is a slight decrease between, now this was a little anomalous. I say increase, decrease, and then something which is abnormal. So slight decrease between 2009 and 2010. Then the third mark scheme point was proportion of women receiving ART increases. Now that's a trend. And then if you gave me a data to support either mark scheme point 1 or mark scheme point 2. So if you gave me data that this and this data from the graph, well, you got a mark for that. So there was a 3 mark, and you have to give me more stuff which is actually given in the question. Now in this situation it says suggest and explain the global importance of providing ART to all pregnant and breastfeeding women living with HIV. HIV can be passed from mother to baby, so if you give them ART, well, that could save the mother and the child, so reduces the number of women dying from the disease, and then the child may grow up without the HIV, and the healthy women can contribute to the workforce or be the parent looking after the child, can be the main carer if partner has died. So overall financial, the child will not be orphaned, and there'll be financial savings, and the mother can work. So if she's getting ART, she is a very important person in society and helping her child to grow up. State what is being injected into the mouse in step 1. Of course, I've skipped all those points because I want to just finish this question. So state what is being injected into the mouse in step 1 is an antigen. Why several weeks rather than several days separate step 1 and step 2. Immune response can occur. And state one feature of the myeloma mouse cells used to step 3 that is essential for the production process. They are immortal. Cancer cells are immortal. They're long-lived, able to reproduce, cell division take place, and they can survive in cell cultures. Name the fused cell. This will be a hybridoma. Last is suggest why step 6 is necessary. Naturally, you want to check the cells which are producing the antibodies. You don't want the cells which are not producing the antibodies. So only those cells need to be cultured, because what we want is the antibodies. So we are not going to place all the cells in the nutrient. We are only going to select the ones which are actually the ones which are producing the antibodies. So you check the cells for the production of antibodies, and only then those cells will be cultured. Okay, now that finishes these 4 questions of the paper 2-2, and we will try to finish the remaining in the next video. So thank you very much for now, and I will be uploading this for all the AS students taking the AS exam, the paper 2 exam, and thank you very much.